Okay, good morning, viewers at home. My name is Adeotunde Michael. I am here again as the team leader of Omega Bio-Edutainment. Don't forget that at Omega Bio-Edutainment, our focus is mainly on three. First one being education, the second one entertainment, the third one is entrepreneurship. We also have some other things, thematic areas in this bigger Bio-Edutainment. And today, on our educational series to this morning, we are going over before now. In my earlier video, I actually spoke at length on how genes can be expressed and what are the functions of genes that will be expressed, what are the components of expression of a gene, how is a gene that is being expressed or information that is being expressed in prokaryotic organism, how is that actually different from the one that is being expressed by the prokaryotic organisms. And in the earlier video, just like I said, we just made mention of gene expression in form of just some passing statements. And today we want to actually re-examine it and look at our understanding, re-examine our understanding on gene expression and how gene can be regulated as well. Don't forget that we are actually interested in a gene to be a set of information whenever you see a bacterial cell that is virulent. Let's say this is a bacterial cell that is actually virulent. Before this bacteria will decide on actually what to do, bacterial cells do not actually depend on communication in form of other thing apart from being a kind of a signal. So bacteria depend strictly on signal. And don't I want you to I want you to remember that the nucleic acid is the informational molecule, just like I said, and as a kind of what you should know as a student of molecular biology, you should know that how is information being expressed, and how can it be actually interpreted at the other end of a bacterial cell. Don't forget that I told you that uh, the first thing is to look at the structure of a gene and as well examine it in form of how information are coded and how can it be translated from one molecule to another. Just like you know at this level that DNA is a double-stranded molecule, and as that DNA being a double-stranded molecule, it is going to be transcribed, it's going to undergo the process of transcription, okay, process of transcription, and this is going to be transcribed into a single-stranded RNA, and in this form it is going to be messenger RNA. It is going to be messenger RNA. Don't forget that. So before we actually so from DNA to RNA, it is known as a forward transcription, because that is coming from double-stranded to single-stranded. It is called forward transcription. Whereas from because you have it in some retroviruses, that is RNA viruses. So this is going to undergo a kind of a reverse transcription, and the enzyme that actually characterize that is a reverse RNA polymerase. RNA polymerase. So from this RNA, it is going to form a kind of a polypeptide. And polypeptide is actually talking about protein. So, and the process of that is going to be through the process of translation. This is exactly what is known as central dogma theory. So, what do we actually mean by gene expression? Gene expression is a process of expressing expressing of the information from one molecule to another molecule, that is from DNA to RNA. And whenever this one is done, the overall product is going to be a functional product. It must be a functional product. So that is exactly what is called gene expression. Now, apart from this forward reaction in which gene is being expressed, there are step-by-step levels at which the gene that will be expressed is actually regulated. It's just like when you switch on a when you switch on a bulb and you do not put it on, your energy is going to get wasted if it is just switch on indefinitely. So bacterial cell or prokaryotic organism or any organism cannot actually do that. So it must be regulated. So this topic now, that is why we are going to examine how gene is being expressed and how can it be actually controlled or regulated. So don't forget that I said this course is molecular genetics, and with a step-by-step focus on step by step of how genes are expressed and how can it be regulated. So regulated, okay, gene regulation. So just like I said that any information, so let's try and look at this. Let's imagine that this is a typical strand of DNA, which is double stranded. So we have a DNA that is double stranded. Let's assume that this one is a G, this place is T, this place is A. Let's say this place is C, this place is A. If this place is T, if this place is G. So don't forget. So this place, according to Chargaff rule, based pairing principle. So this is going to be triply bonded with G. So this is going to be T, this is going to be A, this is going to be C, this is going to be T, this is going to be A, and this is going to be C. So now, okay, in furtherance of that, let's see if we have G, C, A, T. So this one is going to be C, C, G, C, A. Okay, so that what we now have, so that if this plane, you don't forget the theory of anti-parallelism. So this is a 5 prime to 3 prime, and this is going to be 3 prime to 5 prime. 3 prime to 5 prime. So the process of transcription, it's going to proceed from 5 prime end to 3 prime end. So that is why, so now during the process of what is called transcription, so what do we call transcription? Transcription um, is just a process whereby the copy of DNA to RNA, the copy of DNA is made into single-stranded RNA, and the type of RNA that this is going to produce RNA. And just like I said, that transcription proceed from 5 prime end to 3 prime end. So that will now leave us with a strand of RNA, and it is going to be 5 prime. I said it's going to be 5 prime. So it's going to be G, T, A, C, A, T, G. Then this is going to be G, T, A, T. So this is going to be the transcript. It's going to be mRNA transcript. So it is messenger RNA. And the process of transcription actually proceeds in 4 different stages. The first stage is called initiation. Okay. The second stage is elongation. The third stage is called termination, whereas the last stage is called post-transcriptional control or post-transcriptional modification. IETP. I is standing for initiation, E is standing for elongation, and T is standing for termination, whereas P is standing for post-transcriptional modification or transcriptional control. Okay, now you need to ask yourself, what are the enzymes that will be involved in transcribing DNA molecule to mRNA molecule? There are basically, depending on whether it is a prokaryotic gene expression or it is going to be eukaryotic. It is prokaryotic. If it is the enzyme that is involved in prokaryotic transcription is basically RNA polymerase. That is in prokaryotic organisms. Okay. Then in eukaryotic organism, there are basically 3 types of RNA polymerase. So we have RNA pol 1, we have RNA pol 2, and we have RNA pol 3 if it is in eukaryotic organisms. And what each of these does is that RNA polymerase 1 will synthesize ribosomal RNA. RNA polymerase 1 synthesizes ribosomal RNA. RNA polymerase 2 will produce messenger RNA. Then RNA polymerase 3 will now produce transfer RNA. These are typical involved in the translational principle of converting messenger RNA or RNA into protein. We are going to see all this as we actually go on. And don't forget that I told you that there are 4 basic stages of transcription. The first stage is initiation method, initiation step. The second stage is elongation step, and the third is termination stage, with the last being post transcriptional modification. Post transcriptional modification. Okay. Now, let's move forward. Let's move forward by considering the activities of transcription. Don't forget that the first one is initiation. The first step is initiation. Now, whenever you draw a messenger RNA or gene regulation, or the gene of a bacteria or this. So the first thing is that, the first thing that will be involved at initiation method will be number one. Initiation method number one is that transcriptional factor TF, and this have TF2, we have 3 and 4. And just like that, transcriptional factors will bind at promoter region. It is going to bind at promoter region, and by the time it binds to promoter region, so it binds or binding at promoter region. So after the binding, this transcriptional factor is going to unwind. It is going to unwind the DNA molecule. Well, don't forget that the DNA molecule is double-stranded. It is going to unwind it. That is, it's going to separate it at that level. Now, on the promoter region, there is a region that is called Tata box. Tata box. This is basically one of the components of the RNA transcript. Okay, and this Tata region consists of this Tata box or Tata region consists of start from -10 because it consists of about 10 sequences. So that is a Tata region. So, and it is the Tata region is going to be the point that is going to initiate the process of DNA transcription. Is the one that is going to do. So, in prokaryotic organism, in prokaryotic cell, we have 10 sequences, whereas in eukaryotic, it is made up of like 70 sequences. It's made up of -70 sequences, whereas this Tata region in eukaryotic organism is going to start from -35 instead of from -10. So that is very, very important. You should know, just like I said, that the Tata box is a region that is a box or is a region at which the translational factors is going to get it initiated. It's just like that is the decision-making center. That is the decision-making center of transcription, because it is at that point in time, and it is at that point that decision will be made by prokaryotic cell or eukaryotic cell to undergo the process of transcription. At this level, don't forget that immediately this Tata box has initiated it, the transcriptional factors which depend on the subunit, the transcriptional factor is going to bind at the promoter region of the cell. of the prokaryotic cell is going to bind at that promoter region. Then after that, after the binding, there will be unwinding. That is, the two strands of DNA will become unwinded and it will become separated. So once it becomes separated now, yes, once it becomes separated now, it is going to be transcribed into messenger RNA. And I told you that transcription from DNA to RNA is such that if it is a prokaryotic cell, it is going to involve RNA polymerase. But if it is a eukaryotic, the RNA polymerase 1 will give rise to ribosomal RNA. That of pole 2 will give rise to messenger RNA transcript and transfer RNA. But don't forget that in one of the major differences between RNA and DNA is such that in RNA you do not have T. The T is already being replaced by another nucleotide base, which is uracil. So wherever you see T on the DNA, it will be replaced by U. So you need to take note of that. Okay, and that is the first step of initiation. So it's going to unwind. Why is it unwinding it? So as to expose the basis. For further processes of transcription. So it is at that point that you have that. Then again, this RNA polymerase also has a specific point or specific region that it's going to bind to. It's not going to just bind anyhow. It has a specific region that it's going to bind to, and that region, there are 4 different regions, okay, at which the RNA polymerase can begin to act. Don't forget that as an enzyme, that enzyme is going to have an active site at which a substrate is going to bind to before it can become effective. So this is actually going to involve 4. So it's just like that region is like, it's going to be like this, okay, and this is what is called the core enzyme. And this core enzyme will bind to sigma units. It's going to bind to sigma units, and whenever there is binding between them, it is going to produce what is called holoenzyme. So this enzyme will become so activated or so agitated to actually proceed in that process of transcription. Don't forget one of the reasons why we have involvement of enzymes in biological cell is just because most of biological processes cannot proceed without enzymes. Just like the normal chemical reaction cannot proceed without catalyst. So the same thing. Now, after this, don't forget the Tata box. The transitional factors will bind at promoter region. The Tata box is just the initiation thinking center that, oh, there should be they should proceed on transcription. So immediately after that, the Tata box, I said in prokaryotic cell, will start from -10. That is, there are about 10 different nucleotides. So, and 4 of it is what they call Tata box, because that is the arrangement on the promoter region. So, but if it is in eukaryotic organism or eukaryotic cell, the starter box, the promoter region is made up of more than 70 nucleotide bases, and it is at -35, exactly -35 sequence that the transcriptional factor is going to bind to. So immediately after that, the next thing that will take place is unwinding of the double strand of the DNA. And that unwinding, don't forget that I told you that transcription proceeds from 5 prime to 3 prime. So even if you have any any DNA strand like this, it is only the strand that starts with 5 prime. It is only that one that is going to proceed in that other region from top like this. For example, if this one is going to be involved, so that means it's going to start from bottom, because it is transcription always proceed from 5 prime end to 3 prime end. Okay, so the next thing after the initiation step, okay, then I told you that RNA polymerase will not just bind at any point. It is going to bind at a place that is made up of core enzymes, and these core enzymes are made up of 4 subunits. The first one is called, we have alpha subunits, we have beta subunits, we have sigma subunits, and we also have real subunits. So the enzyme will not just bind at any point, it is going to bind at the sigma subunit, and whenever that one has binded, it is going to produce a product that is called holo enzymes. So if this is now formed, these holo enzymes, the confirmation that holo enzymes has been formed is going to, there are going to be bubbles. Production of bubbles will tell us that that is initiation step, and holo enzymes has been formed. Okay, so okay, I said the double-stranded will be exposed by that RNA polymerase. And why is it exposing it? So as to make sure that this strand will actually have a place to bind to. It's going to expose all the genes that will needed to be transcribed. So transcription is just like a process of having a new strand of DNA, a new strand of messenger RNA formed from double-stranded DNA. Okay, so now the next step after initiation is elongation. Okay, the first step is initiation, and there are, you can actually talk of about 3 or 4 or activities that will take place at initiation step. So the next one is elongation stage, the stage of elongation. So what actually happened at this level? Okay, what actually happened at this level is that the RNA polymerase is going to form a complementary strand. Don't forget, it is the RNA polymerase that has unwinded the DNA, and after unwinding it, it's not just going to unwind it just for unwinding sake. It is going to unwind it because of certain reason, and that certain reason is exactly the second stage of transcription, and that is elongation. So there's going to be synthesis of complementary strands, so that now initially you have GUA, CAU, GC, GGC, and AT. So the complementary strand to that one is going to be what? It's going to be CAU, GUA, and CCGUA. So UA. So this is going to be the complementary strand synthesized from the templates of DNA. Okay. Now, what is actually involved? So this RNA polymerase must have read that